Welcome, everyone. This is the first Research Ethics and Policy Series lecture of 2024. It's great to see you in the room, and hi to everyone online. My name is Holly Fernandez-Lynch. I'm an assistant professor of medical ethics in the Department of Medical Ethics and Health Policy. I'm the REPS co-chair along with our department chair, Steve Jaffe, and we are thrilled today to welcome Dr. Jonathan Kimmelman, who's visiting the department for most of January. His lecture today is called Assessing the Scientific Case for Early Phase Trials and/or Innovative Care, the PATH Approach. I'll introduce you in just a moment. A couple of quick announcements. Next month, I'm on the REPS docket, so I'll be host and lecturer presenting on some of my work on the ethics of prioritizing clinical trials at the research site level. You can see our full list of spring 2024 REPS speakers on the slide, and they're also all posted online. As we continue to add to our lengthy list of prospective REPS speakers, we encourage you to share suggestions that we should consider, especially individuals who come from diverse backgrounds or who would contribute diverse perspectives to issues relevant to research ethics and policy. Just a quick reminder of some online resources from our department. We have a series of short research ethics courses available for free continuing education credit, a few of which were recently updated, some minor changes. And these can be used to satisfy research ethics training requirements at Penn and CHOP. Thank you to our many REPS co-sponsors who help make the series possible. And of course, to our REPS coordinator, Lisa Bailey, for making sure everything moves along smoothly. Okay. So for the main event, we have Dr. Jonathan Kimmelman, who is a James McGill professor in the biomedical ethics unit at McGill University in Montreal, where he directs the group for Studies of Translation, Ethics, and Medicine, or STREAM, alongside a variety of other groups with great acronyms. You must be good at coming up with those. Jonathan holds a PhD in molecular biophysics and biochemistry. He has cross appointments in experimental medicine, epidemiology, biostats, and occupational health, as well as human genetics. His research revolves around the ethical, social, and policy dimensions of translational research. And his current projects include investigations of risk, prediction, validity, and knowledge value across the trajectory of drug development. He served in an advisory capacity to the World Medical Association, the FDA, and the Canadian Institute of Health Research. He currently serves as a deputy editor at Clinical Trials and an associate editor at PLOS Biology. And I can personally attest that he is an excellent and thorough peer reviewer. Send him your papers for comments. So just a quick word about how we will handle questions and discussion. When the lecture concludes, I'll ask first in the room for any questions, and we'll kind of ping pong back and forth to those of you online. We are recording this session, and it will be posted to the department's website as soon as it's ready. And with that, Jonathan.
Thanks very much for the introduction. It's a personal thrill to be here. First of all, this Richards building is just about my favorite architectural, accomplishment in Philadelphia for sure, perhaps even in the world. I'm a big fan of Louis Kahn. The other thing that's personally meaningful here is that I actually think that before this room was renovated, I took a summer course in cell biology as an undergraduate here. And so it's really a thrill to be back in this room, not as a student, but as someone that's presenting my research to my colleagues. Also, before I start, I want to thank Holly Lynch for this very generous invitation, Steve Jaffe for the support as well in hosting me for this month, and of course, Lisa Bailey for making this an incredibly easy and seamless process being here. Also, the other administrative support, Emily Walters in particular, for helping with various other administrative details. Okay, good. So let's get into the PATH approach. So part one, the problem. Everyone here knows that drugs are extremely expensive and they are potentially dangerous. Adverse drug reactions account for between the 4th and 6th leading cause of death, at least according to one commonly cited analysis years ago. And what this means is that when we use drugs, it's critical that we have good evidence to suggest that the benefits of using that drug are going to be outweighed or are going to outweigh the risks and the costs of using those drugs. Now, in many arenas in which we're making decisions about the cost, risk, benefit balance for drugs, we're often making those decisions based on high quality evidence, large randomized controlled trials that are sufficiently powered to detect a clearly precise effect. So in particular, the kinds of studies that are used to make these kinds of policy decisions, whether we're talking about regulatory approval or reimbursement, oftentimes are based on strong forms of evidence. They're also based on investigations that use a fairly standard methodology. Clinical trials, although they vary a lot from one to another, they're basically similar in terms of their form. And as a result, there are all sorts of tools available for aggregating the results across multiple trials so that you can at least improve the precision of your estimates around a treatment and perhaps address some of the biases in those various studies. So that's great. But the problem is that in many arenas of medicine, and especially in research, oftentimes the evidence we have available for making decisions is not based on those strong forms of evidence that can easily be aggregated into a meta-analysis. In many arenas, as I'll talk about later, decisions are made on the basis of phase 2 evidence or even just preclinical evidence. In particular, when you're developing a drug, by definition, you often don't have phase 3 evidence available. If you did, you wouldn't be developing the drug. And so at that point, you're often relying on preclinical and or phase 1, phase 2 evidence to make a decision about launching a clinical trial. So just to underscore, for those kinds of settings where we're relying on phase 2 or preclinical evidence, we're talking about forms of evidence that are often fairly weak. For example, they might not use randomization. They're non-standard. If you think about the variety of the kinds of evidence we use to launch an early phase study, ranging from in vitro studies in cell culture through to pharmacodynamic data from clinical trials, there's a wide variety of methodologies. And as a result, there are no simple ways to aggregate or pool results that we can form some kind of a risk-benefit estimate in making a decision, either about advancing a drug into later phase testing, or perhaps using a drug in innovative care, or as I'll talk about a little bit later, reimbursing a drug for which we don't have high quality evidence. Now, this obviously has implications for patients, whether they are patients receiving drugs based on low quality evidence, or whether they're patients participating in early phase clinical trials. But as I alluded to yesterday, it also has implications for research efficiencies. We want to make sure that we are assigning our scarce research resources towards the most promising hypotheses. It's accordingly critical that we have some way to carefully evaluate the evidence before we launch later phase or even early phase clinical trials. So the question is, what's the approach we use of meta-analysis, the standard way of trying to aggregate different findings to form a risk-benefit analysis? If that's not available, what are the alternatives? So before I get to the alternative I want to propose, I want to underscore the exigency of answering this question, because one easy response to what I'm going to propose is that we seem to do a pretty good job at doing this already. Do we really need a formalized method to do this? So I think there are any number of different reasons to believe that the ways that we currently approach risk-benefit analysis in early phase studies and or innovative care, and for that matter, in any other setting where we're relying on low quality evidence, are prone to a number of major deficiencies or risks. Number one, you can point to anecdotal evidence. There are a number of episodes where decisions were made about launching early phase studies that turned out in retrospect not to have been wise decisions. This is, for example, a case in France where a healthy patient, died in a phase 1 clinical trial. A subsequent investigation of that clinical trial by the French regulatory authority found that there actually was not very strong preclinical evidence to support that clinical trial. And they underscored the importance of evaluating preclinical evidence when making decisions about the risk-benefit in early phase clinical trials. There are also any number of other examples one can point to. This is a case of a stem cell study for spinal cord injury, where it turned out in retrospect that there was evidence suggesting the stem cell intervention actually did not work in preclinical models, and yet the company pressed forward into clinical trials of that drug, of that stem cell. Other forms of evidence I can suggest are a little bit more systematic. This is a widely cited study or commentary in Nature, Glenn Begley, Lee Ellis, that reported, so Glenn Begley was a scientist at Amgen, and Amgen, before they start developing a drug, they want to replicate basic science studies to establish that this drug target is in fact a valid drug target. What he reported in this study was that only 11% of the basic science studies that Amgen attempted to replicate withstood replication. One sort of ironic feature, by the way, of the study that I always like to underscore is that they're not exactly clear about what their definition of reproducibility is, nor about how they reproduced. And so ironically, this study in and of itself is not reproducible. But there have been more reproducible methods to assess the reproducibility of preclinical research, particularly in the realm of cancer. For example, in eLife, a couple of years ago, there was a report of an attempt to replicate 17 very high impact findings in cancer biology and cancer preclinical research. And the net upshot of what they found was only about 50% of those 17 high impact findings withstood replication. That is, they led to the same conclusions in the replicated study that you would have had in the original study. So we have reason to think that much of the evidence that's used to support early phase studies is at risk for reproducibility challenges. As well, there are various studies, this is just one example of my own research, suggesting that frequently when phase 2 studies are launched, at least looking at the citations of the phase 2 clinical trial reports, oftentimes, I think I can't remember the exact number. Yeah, okay. Only about 50% of the cases are there citations of preclinical studies suggesting that the drug works in the same indication as the clinical trial. So oftentimes decisions to launch phase 2, phase 1 studies are based on potentially weak evidence. And finally, this is just to underscore that many decisions in medicine are often made based on forms of evidence that are not strong. So these are, for example, NCCN guidelines. There have been any number of analyses looking at the quality of evidence supporting NCCN guidelines. In this study, which has subsequently been followed up and come up with similar numbers, 83% of recommendations in NCCN clinical practice guidelines are based on what they call low-level evidence. Low-level could mean open label studies, it could mean case reports, it could mean subgroup analyses within randomized trials that are supported by expert consensus. Finally, you can point to any number of realms of medicine that deliver interventions that are based on questionable preclinical and or supporting clinical evidence. This is just a slide showing examples of the number of different clinics that offer unproven stem cell-based interventions, at least in 2016, based on Lee Turner and Paul Knopfler's study. Okay, so I've tried to suggest so far that we want to have high quality evidence to support decisions in launching early phase studies or practicing medicine where we don't have high quality evidence, but we are presented with a challenge of there being no way of aggregating or synthesizing the highly heterogeneous evidence that supports decision-making in those realms. So by way of trying to propose an approach, I first want to lay out a set of desiderata through which that this approach, an approach ought to address. And thankfully, the Belmont report has a wonderful paragraph, not widely read or cited, that describes what a proper risk-benefit analysis should look like. Those of you who are interested in this, I recommend going back to the Belmont and reading this amazing paragraph. They articulate what I at least identify as 5 different criteria that makes for an appropriate ethical analysis of risk and benefit. And I think these are the desiderata that are guiding the approach that I'm going to propose today. The first is that the approach needs to be systematic. What does that mean? Well, it means that there are a lot of different parts that articulate with each other in some kind of coherent way. Number 2, the approach should be non-arbitrary. That is, it should be based on a set of reasons and a set of evidence, not just what day of the week it is or what you had for lunch earlier that day. Number 3, a risk-benefit analysis ought to be based on a totality of evidence. It shouldn't be based on just a narrow sampling of the evidence. Number 4, it ought to be explicit. Why is explicitness important? Well, because people often disagree about risk and benefit. if you are explicit, people can at least identify the source of disagreement and then begin to gather evidence or further information about where that source of disagreement is so they can resolve that form of disagreement. Also, explicitness is key for accountability. And finally, you want a risk-benefit analysis that's going to be accurate. Risks involve probabilities. Those probabilities can be accurate or they can be inaccurate. So all of these are characteristics that you want in some kind of method of analyzing risk and benefit and of pooling the information you have available for early phase studies. So with that in mind, let's consider some of the kinds of approaches that are already used by IRBs and by other decision-making authorities in this setting. So the default approach, at least in launching early phase studies, is that if you're on an IRB or a merit review committee, you're presented with an investigator brochure or a clinical trial protocol that has what to me looks like a wall of text describing all the different reasons why they think this drug is promising and why you ought to approve the proposal. Now, there are a lot of liabilities with the way that these arguments are presented within protocols and investigational brochures. The principal one being that they have a narrative structure. Now, as most of us know, probably from studying medicine and evidence-based medicine, narrative structures are seductive. They often lead to, omit certain details and lead to certain kinds of conclusions that would not necessarily be the case if the information and evidence was presented in a systematic manner. And in fact, my group has done one study. It's one study, so it has not been replicated, where we actually randomized experts to look at systematic review evidence versus narrative review evidence from an actual clinical trial that was underway at the time, and asked them to estimate the risk-benefit balance as well as to predict the results of the clinical trials. And what we found in this case was that when people read the systematic review evidence, they were much more pessimistic about the trial being positive. And it turned out when the trial was complete, they actually were also more accurate in estimating the outcome of that trial than reading the narrative review. Now, the other problem with the ways that data are presented in protocols is that they're often missing key elements that you would need to assess the strength of support for an early phase study. This is a study that was led by Daniel Streck. I'm on this paper, but Daniel and Suzanne led this effort, where we looked at the fraction of investigational brochures that included any information about the risk of bias of preclinical studies used to support early phase clinical trials at a German university. And the crux of what we found was that only 5% of investigative brochures had any information about whether randomization or blinding or power calculations or any of these other kinds of measures for controlling internal validity were implemented in preclinical studies. So not crazy about the approach that we currently use. What about other proposals that people have made about reviewing preclinical evidence and supporting evidence? There's a number of different teams that have proposed quality rating approaches. Camarades in Scotland is one of these teams. There's another team in the Netherlands. Basically, what they do is they lay out a set of criteria or score criteria that you use to apply to preclinical studies. The problem is that they're focused entirely on only one type of evidence, namely studies in live, non-human animals. Whereas, as I'll describe later, the kind of evidence that's presented in clinical trial protocols is highly variable and takes many different forms. Number two, they're focused on a very narrow set of validity threats, in particular internal validity threats, as opposed to construct or external validity threats. These kinds of approaches tend to be focused much more on reporting as opposed to the actual content of what was done. And it's very unclear in these approaches about how to combine different information from preclinical studies with in vitro studies, with clinical trials and whatnot. So I think limited applicability in terms of these quality rating approaches. Now, there also are a number of teams that have approached this by proposing checklists and various workflows that you would use before you launch an early phase clinical trial. This is just one example from a group, I think, in the Netherlands. And it's an interesting approach. It's useful in certain ways, but once again, they're focused on a fairly narrow set of validity threats, mainly internal validity threats. There's a fairly narrow definition of evidence. Again, I want to emphasize the importance of capturing the totality of evidence. And many of these proposals are really focused on only one aspect of the decision. In this case, this particular guideline is focused on what the starting dose is for an early phase study. But I want to talk a little bit more about this narrow definition of evidence. So what kind of evidence are we typically using to launch an early phase study or to provide innovative care, or in some cases to practice medicine when we don't have high quality evidence? Oftentimes, the kind of evidence that people are appealing to involve preclinical studies in animals. They could be basic science studies. They could be in vitro studies, pharmacology studies in humans, as well as non-human animals. And often, to a large degree, there is an appeal to clinical evidence. There could be early phase trial evidence. There could be trials of similar drugs in different populations. So we have a lot of different forms of evidence that need to be pulled together to make a decision about launching an early phase clinical trial. Now, the key to thinking about ways to pool those kinds of findings and to formulate a defensible position about the risk and benefit is to abandon the typical mindset that we use when we try to pull data in other realms of clinical, in other realms of medicine. So the standard approach that most of us are used to, meta-analysis, has what I call a molar mindset. The idea is with molar causation is you've got some treatment, you give a drug, you have no idea what's going on inside the body after you deliver the drug. All you know is you get some kind of outcome that's a good outcome, and you can establish a cause and effect relationship between giving the drug and getting this kind of outcome. And it's really this molar mindset that is what's driving not only the design of clinical trials, but also the approach that we use in meta-analysis. If you look at the kinds of information that is typically presented in clinical trial protocols, what you will find is that there isn't actually a lot of molar causal information within clinical trial protocols. Oftentimes you see elaborate descriptions of pathophysiology, molecular biomarkers. There's oftentimes claims that are made based on surrogate endpoints. There's all sorts of functional assays, et cetera, et cetera. What's really going on in any kind of proposal to launch an early phase study is a mechanistic argument, not a molar argument. An argument that you can give A will affect some intermediary pathway, that intermediary pathway will affect some other intermediary pathway, and it'll eventually cause what you want. You'll get to that maybe molar effect through a mechanistic pathway. And that is really, I think, key to, that sort of mechanistic mindset is key to devising an approach to evaluating risk benefit in early phase studies, innovative care, et cetera, et cetera. And that's what I'm proposing with what I call the PATH approach. So PATH stands for Preclinical Assessment of Translation to Humans. So yes, this is another one of these acronyms. If you're in medicine long enough, you pick up the importance of coming up with pithy and memorable acronyms. This is a useful one. And I want to underscore two things before I get into a description of PATH. First, I want to underscore my indebtedness to the work of Steve Goodman, as well as Jonathan Williamson, a philosopher of science in the UK, and a number of other people who have proposed similar kinds of approaches. So what I'm going to propose here is kind of building on some of the theoretical work and some of the practical work that those individuals, as well as a few other individuals, have done. Secondly, I also want to state that in developing the PATH approach, this has not yet been peer-reviewed, but we did send out our PATH proposal for pre-review to these different individuals to get their feedback and to incorporate it into our PATH approach before we submitted it for publication. So I just want to come clean. I want to sort of explain that this has been pre-reviewed. However, the pre-review should not be interpreted as endorsement. I'm trying to underscore there is a quality control step in here, maybe an imperfect one, but it's there. Okay, so before I get into PATH, I want to start by giving kind of a model scenario that's going to I'm going to use to kind of unfold how PATH approach works. So first, I'm going to refer to the target scenario. So this is the idea that we've got some drug. We want to use it in some particular patient population, let's say in this case, glioma, in order to get some kind of an outcome survival. That's the ultimate goal that we want for this drug. Maybe not for the trial, but our ultimate goal with developing this drug is to have a drug that will extend survival against glioma. And in this case, I'm just going to make up a name of a drug, B101. So I want to sort of describe how you might present the evidence for achieving this target scenario using the PATH approach. The basic premise of the PATH approach is that there are essentially 9, you can sort of parse the mechanistic steps of affecting the clinical outcome in the target scenario into 9 mechanistic steps. Now, I'm going to explain these in a second. First, I just want you to note that on the left-hand side, there is direct evidence. This is evidence that you might gather from the actual target scenario, so from clinical trials of patients with glioma. In particular, their goal in developing a drug, again, is to get this molar effect. You give the drug, you get some kind of a clinical effect. In this case, we're giving a drug for patients with advanced glioma, and the clinical effect we're trying to achieve is some extended survival. But in order to obtain that clinical effect, we have to get through various mechanistic steps. So, for example, the drug has to hit its target, in this case, BRAF inhibition. I'm just making this stuff up for the example. The molecular mechanism needs to lead to some kind of physiological change, tumor shrinkage, and that physiological change needs to lead to some kind of desired clinical effect, survival. Now, at the point where you're launching an early phase trial or practicing innovative care, you will often have some clinical evidence from the target scenario on which to make that, to support that claim. So, for example, if you're running a phase 2 trial, you might have support from a phase 1 trial with pharmacodynamic analyses showing that you can actually hit the target with your drug. Or you might have a patient that has glioma in your mixed malignancy phase 1 study, and that patient showed tumor shrinkage. And so you have some support that you can affect the physiological mechanism. Or you might have meta-analyses suggesting some relationship between tumor shrinkage in glioma patients and extended survival. But oftentimes that evidence is going to be weak. It's going to be based on a small number of patients, non-randomized studies, et cetera, et cetera. And that means that in order to make that claim, to substantiate the claim that you are going to achieve your target scenario, you're going to have to appeal to the right-hand side of the path diagram, the model evidence. And in particular, there are two kinds of mechanistic steps that you're going to have to appeal to. The first are mechanistic steps showing cause and effect relationships. So, for example, this might take the form of biochemistry studies showing drug inhibition, or it could be in vitro experiments showing drug action, could be any number of different kinds of experiments showing that you can move vertically in the path diagram. So, for example, here's one type of evidence you might see presented in a protocol. In cell culture, they give B101 and they see BRAF inhibition in vitro. That could be used to substantiate the idea that in a model system, you can get from delivering the treatment to modulating your target. Now, the second form of evidence, that horizontal form of evidence, is translational. Basically, what that evidence is going to suggest is that if you affect these outcomes in your model scenario, that is going to be in some way predictive of what you'll see in your target scenario. So that might come from meta-analyses showing some relationship between outcomes in models and outcomes in patients. Or it could come from some basic science showing that the mechanism that drives disease in patients is recapitulated completely or almost completely in your animal model system, or various other forms of evidence that you're using to substantiate the translational relevance of that model outcome. So again, here's an example of a kind of vertical evidence. You give B101 to glioma xenograft mice, you get some kind of outcome, tumor shrinkage, and next you need to substantiate the translational relevance. And again, that might come from meta-analyses or again, evidence that your xenograft mice recapitulate the same mechanism of glioma progression as what occurs in patients. Okay, so these are the 9 different steps of the path approach. And what's key in path is that you can form at least one path of mechanistic evidence that gets you from delivering the drug, either in your target scenario or in models, through to the clinical effect down in the lower left-hand corner. So how does path actually work? How would it be applied? There are essentially 3 steps in the application of path. The first step is to gather or assign evidence to steps. I emphasize gather because if you're designing a protocol, you would go about trying to do literature searches to gather the evidence to support that you can affect each one of these different steps. If you're an IRB or a merit review committee, your task is to take what's written in the protocol and to assign it to each of these different steps. So, for example, you come across a statement like this: B101 is an avid inhibitor of BRAF with an IC50 of 0.1 nanomolar. Okay, so that's a statement that belongs to this position in the path diagram. Here's another kind of a statement about xenograft mice and their tumor shrinkage that can be assigned to this step. Now, what's key, again, is that when I use the word model, I just mean your non-target system. So, for example, in that case that I described, we're talking about non-human animals as a model. Patients can also be a model if they are patients with your non-target disease. Patients with colorectal cancer, who responded to this drug could be invoked as evidence that your drug is going to be useful in glioma patients. So I just want to underscore that even though you're talking about patient evidence, that is essentially another form of model evidence that you use to move vertically on that right-hand side of the path diagram. Now, as I stated earlier, in order to make the claim about the translational relevance of that model evidence, you have to substantiate that there is some predictive value or predictive relationship between the outcomes in that model system and the outcomes in what you expect to see in your target system. So here there are various hints that are provided in a description of the vertical evidence that would help a reviewer. In this case, it would have to be a very savvy and well-informed reviewer to formulate some opinion about the translational relevance of that information. And that could be assigned to that particular step. Okay, so that's same thing with clinical evidence. So that's the first step. You have a series of different bits of evidence that are invoked in a clinical trial protocol. They're assigned to each of the different positions within a path diagram. The second step would be then to assess or to describe the strength of evidence at each one of those positions in the path diagram. Now here, I'm going to lean heavily on the grade criteria and invoke three elements that you would consider in the strength of evidence. First would be the magnitude of the effect. The second would be the precision around that effect estimate. And the third would be the risk of bias or the internal validity of that effect. Certainly for these kinds of vertical arrows, these are going to be directly applicable. A statement like this, kind of thing that you would pull out of protocol, is providing at least some information about the precision. I think in this statement, they mean statistically significantly reduced tumor growth. There's no information in this sentence here about magnitude or risk of bias. So again, protocols often omit this kind of information. It would be key to forming an assessment, but there's no reason why they couldn't insert information about magnitude or risk of bias into a statement like this so that you can assess the strength of evidence for that particular claim. Now, with translational evidence, it's much harder to assess the strength of evidence, but I think the appropriate way to approach this is to think of this more in terms of a diagnostic, where you're thinking more about the sensitivity and the specificity of your model system in terms of its predictive value for the clinical scenarios that you are envisioning. And oftentimes, descriptions of evidence supporting a clinical trial will contain some clues that at least a very savvy and well-informed reviewer might be able to use to infer something about the sensitivity and the specificity. So in this case, xenograft mice, everyone knows they're a super sensitive, or at least people that work in cancer know they're a super sensitive system, but you get a little bit more specificity if you're using orthotopic implantation, et cetera, et cetera. So step 3 of the path approach now is to combine those judgments into some judgment about the strength of evidence to support the early phase clinical trial. So in this case that I've described, there are several steps for which there is no evidence invoked within the clinical trial protocol. So you can now assign, in this case, you can color code each of those different steps. In this case, where there's no evidence before, they get red arrows, so no information at all. But the other steps, I'm just, again, arbitrarily making up these color codes. You have some strength of evidence to support the claim of clinical effect. Now, what I want to underscore is that there is nothing in the path approach that says that every step needs to be accounted for. Again, the goal of path is to establish the strength of evidence that there is at least one, preferably more than one, path from giving the drug either in a model or in a clinical scenario to get the kind of clinical effect you intend. And in this hypothetical scenario, we have at least 2 paths. Now, what follows afterwards is, an ethical assessment about the risk-benefit balance. I'm not going to talk about that very much. I'll just say really quickly that generally you would demand a stronger support for the trial where you're talking about high-risk treatments, where patients have lots of viable options available to them, or where you have a lot of other drugs that are under development. Okay, so I want to move into sort of unpacking a little bit about the implications of the path approach I've just described. So first, I want to underscore that I think it meets the 5 desiderata that I established at the outset of this presentation. It's systematic insofar as you are connecting the various dots of mechanistic evidence to make some kind of a clinical claim. It aims to be non-arbitrary, that is, it aims to invoke evidence and a set of reasons for having a particular judgment about risk and benefit. It aims to be to capture the totality of evidence. I'm not just focused on clinical trial evidence. I'm not just focused on preclinical studies. I'm capturing everything from in vitro studies to pharmacodynamic studies in patients. It's explicit. You can look at that path diagram and say, you put a red here. I think this ought to be an orange, or I think this ought to be a yellow. Let's talk about that a little bit. So you can isolate where sources of disagreement might be. And finally, it aims to support judgments that are not going to be overly affected by a narrative structure, but instead that are going to reflect accurate risk-benefit judgments. And again, the key to achieving that is applying a mechanistic approach as opposed to a molar approach to assessing risk and benefit. I also think that this approach overcomes a lot of the limitations of the standard approaches I've described that are often used to assess risk-benefit in early phase studies. For example, in the approach that I've described, the validity threats are not just internal validity threats, but I'm also addressing construct and/or external validity threats. But of course, this approach has a lot of limitations, and it's important to consider these limitations either as opportunities to refine this approach or bound the ways that we apply this approach. And I want to describe 3 limitations in particular. The first one that probably everyone in this room is thinking is, that sounds like a great idea, Kimmelman, but completely useless for an IRB given the way we work. Completely unfeasible. Okay, so I actually think there's a way to approach this. I can't think of any reasons why we couldn't, at least in principle, ask investigators when they put forward proposals for early phase studies in merit review or in ethical review or in funding competitions, et cetera, to restructure their protocols so that they abandon that narrative structure. They abandon the way they organize or disorganize information. I mean, by the way, just a task for those of you who look at early phase trials protocols. Try giving them a read, just like think of yourself just as if you were teaching how to write like compositionally, like how to write something clearly. Try giving them a read just purely from that angle, and you'll see that there's just a very strange way that many protocols, not all of them, structure the information. So, okay, so I think that we can actually sort of, encourage investigators to apply this structure when they write the proposal. I also think that in the era of artificial intelligence, it is probably not a lot to expect that there will be various ways of either using AI to construct path diagrams or of using AI to take protocols the way they are written in narrative structure and to convert that into some kind of visualization, where we go from something that looks like this to something that looks like this, where again, you have this path diagram and you have a brief description containing all the information you need for the evaluating the strength of evidence, for the evidence to support that you can get through each one of these different steps. Okay, the second major limitation here has to do with assessing the strength of translational evidence. It is difficult, but not impossible to assess the strength of evidence in those vertical directions. We have all sorts of different ways to do that. We're used to doing that in medicine. Assessing the strength of evidence for the translational relevance of outcomes is extremely difficult, and there are no consensus ways of assessing strength of evidence for external validity. I just want to underscore, this is a chronic problem in medicine. It's a problem also outside of medicine. Economists who are designing randomized controlled trials are constantly struggling with the question of the generalizability and the pragmatism of their economic experiments. So this is just a problem that we deal with anytime we're dealing with science and anytime we're dealing with models. So in order to make this process run, in order to sort of use path profitably, we need some way to index the strength of evidence. I do think this is a difficult but tractable problem that can be addressed. There are a number of interesting efforts underway, both in the clinical trial realm, but also in realms like political science and economics. We can draw from that for indexing the strength of models, or at least the characteristic strengths of those models. And finally, I also think, although I can't establish for sure, that there probably is a structure of justification for model evidence, akin to the kind of model structure that I described with the path diagram. In other words, there's certain kinds of arguments and certain kind of information that must be in place in order to make a claim about the predictive value of a model. So further work needs to be done in sort of uncovering the basic structure of an argument that a system is predictive for some kind of scenario that we're contemplating. Now, the third limitation also is an obvious one. I started off contrasting meta-analysis with this approach. Meta-analysis is quantitative. You get this nice point estimate with these tight confidence intervals. That's great. nothing I've said so far lends itself towards quantitation. So here's how I respond to this limitation. My first question is, do we really need quantification here? I don't know. There's all sorts of realms that we deal with, including in medicine, including a risk-benefit judgment, where there isn't really quantification going on. What we need is a justified set of judgments, not necessarily quantification. But if you insist that we do need some kind of quantification, or if there are particular scenarios, let's say you're dealing with a super high-risk drug or a super edgy clinical trial, I do think there probably are approaches. You'd have to sit down with a bunch of biostatisticians, but I think there are approaches where you could potentially elicit probabilities from a group of experts and combine them mathematically in some kind of a model in order to put probabilities on the outcome expectations for your target scenario. I want to briefly talk about some of the applications of PATH. I've already implied that this is an approach that could be used to assist in merit as well as ethical review of early phase studies and or innovative care. But as I alluded to earlier, I think there are a number of applications that extend beyond this. Another application, I think, is in reimbursement. As we move into a realm of precision medicine or a realm of gene therapy, oftentimes we are going to be presented with questions about whether the evidence is sufficient to support, large, substantial payments for these different kinds of interventions, where we either don't have enough follow-up time to know that about the durability of the effects, or we don't have high-quality randomized controlled trials to convince us that these effects are actually there. In those kinds of settings, in order to make a defensible claim that these are interventions worth reimbursing, you're going to have to appeal to mechanistic evidence that suggests that when you give a gene therapy, its effects last 10 years, even though you don't have trial evidence to suggest that, or mechanistic evidence that suggests that, yeah, we saw one response in a patient, but we expect that when we apply this to other patients with this ultra-rare disorder, we're going to see the exact same effect. The third application here is that I think just in the same way that reporting guidelines like CONSORT that are used for reporting of randomized controlled trials, even though they're reporting guidelines, one of the most important effects has been to create a very strong incentive for researchers to use optimal approaches. So if you know you have to report that you used a substandard method of randomization, now when you publish your clinical trial, CONSORT now, you are sort of are going to contemplate that when you design your trial and use the optimal approach. And I think given that PATH is encouraging researchers to be explicit about the strength of evidence for each one of those steps, it is encouraging scientists to address the various liabilities and the support for their early phase trial or innovative care or whatever it is. And finally, selfishly, I spend most of my time doing research. And of course, this would not be a complete talk if I didn't talk about how many great research opportunities that PATH opens up. So there are any number of different ideas that we are following up on. One of them I'll mention really quickly, a study that's underway now is looking at the prevalence of various kinds of evidence gaps that are in clinical trial protocols. Here, I'll just quickly go through some data. We took a sample of 50 phase 2 cancer trials involving monotherapy, and we quantitated the occurrence of information or evidence supporting each one of the different steps in the PATH diagram. And there are a couple of sort of interesting findings that show you just how kind of gappy clinical trial data often are. So for example, what fraction of clinical trial protocols explain the translational relevance of model systems for the target scenario, at least in terms of explicit statements that say, we use this model because this model recapitulates this disease or it has these following properties that lead us to think it's going to predict clinical outcomes. 0% of animal of protocols contained any explicit translational statements. 2% of protocols, when they invoked model evidence from clinical trials, explained the translational relevance. Now, I want to be clear, there were other statements that a very savvy and well-informed reviewer might use to make some kind of inferences about the translational relevance, but at least in terms of explicit statements that your typical IRB or even merit review member would need, very rarely is this present within clinical trial protocols. Another major gap is that at least when animal evidence is invoked, oftentimes it is invoked absent information you use to assess the strength of that evidence. So for example, only 8% have any information that would allow you to assess the precision of the estimate. on the effect of the drug in non-human animals. Only 27% had information about the risk of bias. Another interesting finding, this kind of surprised me, although maybe in retrospect it shouldn't have surprised me, is that these are all phase 2 monotherapy studies in cancer. Only 46% invoked any kind of evidence from the target scenario. So if you think about this, it's a phase 2 trial, so it was probably supported by phase 1. You would think they would be able to cite phase 1 trial evidence in the particular indication. So typically, in cancer, phase 1 studies oftentimes involve mixed malignancies. So there might be 1 or 2 patients that have glioma in a mixed malignancy study. You would think that they would invoke that evidence to support the phase 2 study. Only about 50% of them invoke any kind of supporting trial evidence in, again, the target patient population with the identical drug. And one other observation, this is something that is kind of expected, but I think it's worth underscoring the importance of this gap in clinical trial protocols. They often invoke evidence that the drug can affect some physiological mechanism, either in the target scenario or in a model, but they rarely provide any information to help the reviewer make an assessment that affecting the physiological change will lead to the desired clinical outcome. So for example, they might say, okay, this drug shrinks tumors in patients or in models, et cetera, but rarely is there any statement saying, we think that tumor shrinkage in this case, there are all the following reasons to think that this will lead to an improvement in survival, which is the ultimate goal of the drug development effort. Now, we can also ask all sorts of other kinds of questions using this path approach. So another kind of question that we're hoping to address is, I started off by saying we ought to expect certain kinds of patterns in the ways that protocols are written. For example, if you have a drug that is a riskier drug, you should expect to see more effort on the part of the trialist to explain the strength of evidence and to invoke various forms of evidence for their clinical trial. Do you actually see that? That's another question that we can address using this path approach. Okay, let me wrap this up by drawing a couple of conclusions. First, again, the sort of take-home messages here. Open up a clinical trial protocol or an innovative care protocol. Oftentimes, you're going to be confronted with information that looks kind of like this. It's deadly, at least in my experience. It's about as well organized as a Jackson Pollock painting. It's very hard to sort of know what aspect of the description to focus on and how to kind of connect all the different components of that evidence to establish a risk-benefit balance. At least it's hard for me. Instead, what we need is an approach that is customized to the kind of problem that we're confronted with in conducting early phase studies, in applying innovative care, or in practicing medicine when we don't have high quality evidence. And in those kind of settings, oftentimes we're going to have to rely on mechanistic evidence. In other words, we're going to have to abandon our desire or our preference to use this kind of molar mindset that we use for making decisions about risk benefit in many realms of medicine and appeal to some systematic way of combining mechanistic information. And that's essentially what PATH is trying to do by parsing the different mechanistic steps into 9 different components. The crux is that you want to establish that when you give the drug in the target scenario to target patients, you will achieve the clinical effect. Much of the evidence for that is going to come from information about what happens in models, whether those are patients or non-human animals, and information about the translational relevance of those different models. I'll close by thanking 3 of my collaborators in this effort: Patrick Kane, a former postdoc, Benjamin Carlisle, a current postdoc / research assistant, and Celine Beiser, a master's student that helped work on this project and really made substantial contributions to this effort. Also, some hat tips to some of the people I mentioned earlier. So Jonathan Williamson, the philosopher of science, Veli-Pekka Parkkinen, also a philosopher of science, Jason Gerson and Steve Goodman, who did some work for ACHRE, I think, that's kind of along these lines. And we've gotten feedback from various other people. Scott Plotkin in particular has given us some feedback on this, and I want to acknowledge their help and support. And I also want to underscore that these should not be read as endorsements of the PATH approach. Finally, I close by just thanking my funders, in this case, CAHR, but also some of this work is supported through a grant from the NIH. Thanks very much.
Okay, so if you're online, feel free to either raise your hand and I can call on you, or put your comment in the chat. Questions in the room? I can start us off, Jonathan, which is to say, it's, I think, really interesting, and I like the idea of using it for merit review and in a tailored way for IRB review. I think it would need some handholding in the protocol as you described it. But I was surprised that you didn't also articulate potential uses for regulatory approval, selection of surrogate endpoints, or, you know, the willingness to accept surrogate endpoints, and maybe even as part of informed consent, not in such a robust way, kind of walking through all of the steps. But the discussion in consent in particular about risks and benefits is often very hand wavy. And my hypothesis is that if you actually like walked people through, here's the evidence that we have and why we think it may or may not work, it would feel in many cases less exciting, right? People might say, oh, I'm actually not so excited to participate in this because it's unlikely to work, or, you know, they may not be as convinced. And so I'm wondering if you could just maybe comment on those other potential uses and the modifications that you think would be needed.
Yeah, I think these are great points. So it is early days in path. This, as I mentioned, this is not yet peer reviewed, so it's submitted right now. And I really envision this as a research program that's going to kind of keep me fueled for another 5 or 10 years. And I've only just started to kind of unpack some of the implications. So you raised 2 other potential applications. The first was about regulatory approval. 100%. I mean, I also think that this is a way to render more explicit, transparent, and accountable regulatory approval decisions, particularly in a realm like precision medicine, orphan diseases, other realms where you're not going to be able to appeal to the high quality evidence. So I would definitely envision trying to expand and extend and perhaps customize this approach for regulatory decisions. I should also mention that I've identified 9 canonical steps in the path diagram, but the idea is that in different kinds of realms, the content of what goes into those 9 different steps might be really different. So for example, in gene therapy versus small molecule drugs, what counts as target engagement, what kind of evidence gets sort of pulled into that. So there's a lot of work to be done, not only to extend this to how a regulator would need to think, but also to extend this to the different kinds of modalities that you would try to apply. So again, in the original work by Steve Goodman and Jason Gerson, you know, they actually did not do this for early phase studies. They did this more in the context of reimbursement, you know, kinds of things. But, you know, they in their argument, this should be applied for surgeries, you know, devices, like, you know, and that's, I aspire to do that as well, although I'm focused initially on the stuff I know best. To the point about informed consent, super interesting idea, something I am thinking about. When I presented this work at a rare disease symposium, I was surprised that both a physician and a patient advocate immediately got up in the question, you know, you think they're going to just shoot this idea down, you know, like, how can you get this into informed consent? Like, how can we use this? And I was surprised because I have always had the assumption that we ought to spare patients the complexity of all this complicated information when we do informed consent. But, you know, I guess their questions and the arguments they made started me thinking about how you would take a path diagram. Right now, it's extremely arcane, the structure, but how you could potentially use this to create an opportunity to have a conversation with a patient who wants to have a conversation to get a sense of the risk benefit. So the NIH grant that I mentioned is a grant that's funding work on early phase neurology trials. And there are interview components where we're interviewing investigators, IRB members, as well as patients. And one of the things we're incorporating into our interviews is a series of questions about whether this kind of information would be useful, so that we can at least begin the process of trying to think about how to adapt this towards informed consent. So that would hopefully be in the renewal.
Great. I have a question from Emily Largent online, who says, I'm very interested in the idea of using this for payers. So many connections to accelerated approval debates in the US. Could you say more about how this relates to cost benefit or other ways of evaluating whether to pay?
So, yeah, I mean, you know, my ideas are not fully fleshed out here, but but at least this is what I'm thinking. I mean, any, claim about reimbursement is going to be based on some estimate of, how large the effect is and how confident you are around around that effect. And so, it's basically the same kind of judgment that you'd be using for paying money versus paying, putting at stake patients by putting into clinical trials. So the basic structure of the problem that you're confronted with reimbursement, even though it's dollars and not people, is the same. You need some sort of an assessment of the strength of evidence. Likewise, when we're talking about, for example, only having evidence on surrogate outcomes, oftentimes, the only way that we can make a guess at the relationship between the surrogate outcomes and the clinical outcomes that we desire and that are grounding the reimbursement decision is going to be based on mechanistic information and evidence. So I alluded to, for example, one approach that you might use is, meta-analyses that show that, tumor shrinkage, correlates with survival. Yeah, that's going to get you kind of part of the way there, but it's not going to get you completely the way there because, in order to, every drug has a different mechanism. And depending on that mechanism, a tumor response might or might not necessarily correlate with improved survival. So, definitely, both at the level of approving drugs, but also at the level of reimbursing drugs, it's going to be kind of a similar reasoning process where, at least at a bare minimum, you are pressed to invoke a set of reasons and a set of evidence to explain why you think this effect on a surrogate outcome is going to correlate with a clinical outcome.
Ask your question.
Sure. Jonathan, this is really interesting and tries to formalize the concept of translation in a way which I find really appealing. In thinking about your the model on, I guess, the left side of your diagram, or over on this side of the screen where you've got your evidence from the model system, whether it's a non-human animal or some other disease, and then you've got your direct target scenario. It was relatively easy for me to understand the vertical steps, like the logic of the vertical steps. And it's also, we talk about arguments why this is a good or a better or worse model for this target condition and appropriate to use or not. But you try to break it down into 3 levels of translation, top, middle, and bottom. And that was where I couldn't quite follow you. What are some examples of an argument that you might make of that translation at the top level, the middle level, and the bottom level, as opposed to just saying this is a good model because X, Y, or Z?
Sure. So, let's sort of go through these. So let's start at the bottom level. You give the drug to some non-human animal and you've done survival curves and the animals survive. So now you need to establish some kind of claim that extending the survival of mice is relevant to extending the survival to humans. Now, at first you might think, well, that's straightforward. Survival is survival. Okay, but let's think about this. I mean, how far out do the survival curves go? Do they go far enough out? What's the comparator that was used? Is it the standard of care comparator, or is it some empty vehicle or whatever? So I think on that bottom, what you would need to establish is what I call the construct validity of the scenario that you used in the model system to establish survival. So at that middle level where I talk about physiological effects, it's sort of a similar logic. You have shrunk tumors in xenograft mice, or you've shrunk tumors in patients with a different cancer, and now you have to kind of explain, well, look, this is a BRAF drug, colorectal cancer is a BRAF-driven cancer. We shrunk drugs in a BRAF, cancer. Because we know, we have, good reason to know that glioma is driven by BRAF mutations as well, we have reasons to think that these results in colorectal cancer are going to be predictive of tumor shrinkage, okay? Now, the top level is a little bit trickier, but the basic idea there is that we give a drug and it engages its target. It inhibits BRAF, in vitro and or in vivo, whatever it is. And there, you rarely will see these kinds of claims made, but you could, in principle, make the argument that we have reason to think that when we engage the target in non-human animals, we're going to get similar engagement in human beings. Here's a scenario where this might occur. Imagine maybe with my drug, B101, in order for that drug to work, it has to cross the blood-brain barrier. So you establish in non-human animal models that this drug crosses the blood-brain barrier, or maybe in tissue culture, any number of different tools. And that is now used to establish a translational relevance that when you give the drug, it's actually going to get to its target.
All right, we are exactly at time. So please join me in thanking Jonathan for a great time.